We will now demonstrate the complete process of fecal sludge sample collection from Chasidol, followed by its transportation and storage at the ENPHO laboratory. The sampling site is situated in Chasidol, Lubu, Ward number 8, Mahalakshmi Municipality, approximately 1 kilometer north of the Lubu bus park. The containment facility is constructed at ground level within the building premises, a location that safeguards it from direct surface runoff while facilitating convenient access for dislodging operations. Tank stores black wastewater generated from 8 members residing in the household. The tank was constructed in 2016. It is a line structure with impermeable side walls with open bottom. The tank measures 2 meter 19 centimeter in length and 1.45 meter in width and has a depth of 5 feet. Since its construction, it has been in continuous use without dislodging. Access to the tank is provided through a manhole fitted with a cover. The researcher begins by arranging all the equipment and materials needed for sampling, such as core sampler, sample container, measuring tape, permanent marker, icebox, tissue paper, measuring cylinder, bucket, and disinfectant spray. Before getting started, researcher puts on PPE such as waterproof apron, gloves, rubber boots, masks, and protective goggles. This ensures safety against splashes of sludge, harmful gases, and pathogens. After this, researcher prepares the core sampler for the sample collection. For this, a metallic rod with an airtight seal at the bottom and a rope at the top is fitted inside a PVC pipe with a diameter of 50 mm. Researcher removes the cover slab of the containment and observes the sludge inside. To determine its depth, researcher carefully lowers a long stick into the containment and then withdraws. The sludge leaves a clear mark on the stick, which is then measured with a measuring tape. The distance from the base of the stick to the mark is found to be 5 cm, which is the depth of the sludge. A sample is collected from this level to ensure it is truly representative. The researcher holds the core sampler device and lowers it through the manhole to the bottom of the tank. After letting it settle for about 30 seconds, the sampler is sealed and removed. The collected sludge is then transferred into a bucket. This process is repeated until the bucket is full. Once enough sludge is collected, researcher homogenizes the sample by mixing it thoroughly. The sludge is transferred into a 10-liter container and a portion of the sample is poured into 1-liter transparent measuring cylinder and allow it to settle. The researcher then repeats the sampling process to collect the next 10 liters of fecal sludge. Type of fecal sludge is determined to be semi-solid and the color is dark brown in color. Finally, once all samples are collected, the researcher carefully cleans and disinfects all the equipment, ensuring everything is ready for safe storage and transport. After collecting the samples, icebox along with necessary materials are loaded in the vehicle for transfer to the ENPHO lab. Lab is approximately 8 km from the sampling site and it takes around 30 minutes to reach the lab. Upon reaching the ENPHO lab, researcher removes the sample containers from the icebox. The sludge is transferred into a 20-liter bucket and mixed thoroughly using a stainless steel ladle to ensure homogeneity. The mixed sample is then divided into 3 parts or duplicates. For the storage of the sample, researcher arranges the different volume of sample bottle based on different type of analysis. For the physiochemical parameters such as total solid, pH, TSS, turbidity, pH, EC, ammonia, sludge volume index, orthophosphate, nitrite, researcher stores the sample in 1-liter bottles without preservatives. pH and conductivity are analyzed immediately, while the remaining parameters are tested later after refrigeration. For the parameters such as COD, TKN, and total phosphorus, samples are preserved with sulfuric acid to keep the pH below 2. And for microbial analysis, researcher stores the sludge sample in the sterilized containers. Throughout the entire process, standard sampling protocols are strictly followed, with careful handling at every stage to ensure accuracy and reliability of the results. This completes the process of fecal sludge sampling, transport, and storage for laboratory analysis.